Coming up next on Passion Struck.
For me, individualizing epigenetics can be done right now and in a very easy way. So we first start from the universal principles. In general, we know that an epigenetically complete diet involves animal foods and plant foods. We know how these foods nourish our epigenome. Then we need to personalize to people preferences and conditions.
Welcome to Passion Struck. Hi, I'm your host, John R. Miles, and on the show, we decipher the secrets, tips, and guidance of the world's most inspiring people and turn their wisdom into practical advice for you and those around you. Our mission is to help you unlock the power of intentionality so that you can become the best version of yourself. If you're new to the show, I offer advice and answer listener questions on Fridays. We have long-form interviews the rest of the week with guests ranging from astronauts to authors, CEOs, creators, innovators, scientists, military leaders, visionaries, and athletes. Now, let's go out there and become Passion Struck. I am so excited today to have Dr. Lucia Aronica on Passion Struck. Welcome, Lucia.
Thank you for having me, John. I'm excited to meet you and your audience today.
Well, thank you. I'm excited for you to be here. And one of my favorite countries I've ever visited has been Italy. And as you and I were talking before you came on the show, I've been there 20 plus times, from Sicily all the way up through northern Italy and everywhere else. And you grew up in Naples. Can you tell me a little bit about how growing up in Naples influenced you and the life that you've led?
Oh, yes. Naples. I'm a proud Neapolitan. Naples is a rough city. It's a complex city, just like biology. It's a mess. But that's why it's so beautiful. And it teaches you to navigate complexity, the complexity of life, and embrace its beauty. It's in Naples where I fall in love with food. And in Italy in general, food is very important. Not only what we eat, but also how we eat it. We also eat it in company, with a family, and we give an important value to the preparation of food. So something striking that I noticed when I moved to the US is that here people value convenience. For example, for many people, it's so cool that you can buy something ready to eat and then just turn on the microwave and it's ready in 5 minutes. To an Italian, that would be so sad. An Italian would rather skip the meal rather than doing something like that, eating something in a box that doesn't have all the magic of the preparation behind it and anticipating the food and eating it with people, or at least taking the time to eat it properly. So this is, I think, something that tells you a lot about how probably Italian people are more resistant to the pressure that food manufacturing are making us believe that something that is quick, easy, it's good for us. Another related thing that is far more common here than in Italy, actually I never seen in Italy, I think I will never see it, is people eating or drinking in the car while they are driving. Every time I see it, I think first this is dangerous, but second is just negating the power of food and taking the time to connect with food. Life is all about connections, whether these are intentional or not, but we connect with the world around us. And food is the ultimate practice of connecting with yourself and your environment around you. You do it every day, multiple times a day, and you train this muscle of connecting you with the environment, exchanging pleasure and cues like learning and being also curious, the food and the flavors in your mouth. I think this is a striking difference from somebody coming from another country, especially Italy, and living here. And I consider myself now an hybrid. So I have some aspects of an Italian and some of an American and an international person. So I think this is fascinating to me.
I remember when I moved to Spain and I was in my early 20s, how different the supermarkets were there. Because as a teen and a young adult, I worked in a lot of supermarkets in the United States. And so much of our food, as you mentioned, is ready to make or has tons of preservatives in it. And what struck me when I lived in southern Spain was the supermarkets were so small, and most of the Spaniards would go every single day to get fresh food. Whereas in the States, we tend to go and get a week or longer duration of food. And just the freshness that you experienced in Spain compared to the way I had grown up was just such a revelation to me. And it's something, as you mentioned, that I saw all throughout my travels in Italy as well. So definitely different cultures and the way that we express ourselves around food. But what I wanted to ask is, growing up in Italy, what led you on this journey to ultimately ending up at Stanford University where you're at today?
It's a long, fascinating journey. First of all, I was a nerd at school since my high school times. So I loved just learning new things. Knowledge was my fuel. And during my high school, I was passionate about literature, philosophy, Latin, and ancient Greek. We were studying in very details, the humanities. And when the time came to choose a university path, I decided to change completely just because I'm so curious. I was like, okay, I've never ventured really, I never deepened my knowledge in the sciences. And at that time, it was the year 2000. And the Human Genome Project, the first draft was just completed. So there was a lot of excitement around the possibility that knowing the sequence of the human genome would reveal lots of secrets about human biology. And I thought, I want to go into this field. Also, my father was a doctor and he died when I was 14. And I am the most rational person in the family. And I felt I didn't want to become a medical doctor because I didn't want to see people get sick and die. The death of my father was a profound event in my life, and I didn't want to experience that anymore. But I wanted to, in a way, follow his path and do something as a scientist in the medical field to benefit people, but from another lens, without having to deal and see sick people every day. I decided to start a program in medical biotechnology. It was a brand new program in Naples. There were only 30 people selected through a test. It felt like being in a high school classroom and I was at the university. It was a very selective program, and most of my classmates at that time are now professors, very famous people, and it was great. And then after my university and master, again, my thirst of knowledge told me Italy is not enough. I don't want to do a doctorate in Italy. I want to do a doctorate abroad. So I applied for several PhD programs in Europe, and I took a year off just to travel through Europe and get interviewed. And then I decided to go to Vienna because, again, they had a position for a PhD in epigenetics, and specifically in an epigenetic mechanism that was discovered only a few years ago, and for which in 2006 the Nobel Prize for Medicine was awarded. The name of this epigenetic mechanism is RNA interference. And this was my first encounter with epigenetics. Since then, I've worked with different epigenetic mechanisms. We can then explain in more detail what epigenetics is and what these mechanisms are. But then I moved from Vienna to Oxford to do my postdoc in another epigenetic mechanism, and then finally here at Stanford, where I'm studying epigenetics in the context of nutrition and how different diets affect this fascinating world of epigenetics.
Well, thank you for that. And the way you and I got interested was from a friend of both of ours, Dr. Kara Fitzgerald. And I remember I was so excited to interview Kara about her book because when it came out, she was explaining a trial that she had done, which was one of its first of its kind, that through natural intervention, she was able to show that she could lower people's biological age. And the crux of what she was studying was the impact of epigenetics on our lifespan and biological age. With that as a backdrop, can you provide an overview of what epigenetics is and why it's so crucial for our understanding of our health and longevity?
Yes, gladly. So the prefix epi means on the top from the Greek language. So epigenetics literally means on the top of genetics. So if our DNA is our genome, then our epigenome is a set of molecular tags of molecules that sit atop our DNA and regulate the expression of genes. So whether they are on or off. Actually, it's more like a modulation than really an on or off switch. And this process of gene expression explains why cells and organisms with the same DNA can look different from each other. Think of identical twins that with time can look different with each other and even develop a different predisposition to disease. Or the caterpillar and the butterfly. They look completely different from each other, but they have the same DNA. It's just that in the process of becoming a butterfly, there are genes that are turned on and off. And even in our body, we have 300 types of different cells, and they all have the same DNA. How it is possible is because we have the same DNA in every single cell of our body, but each of them has a different epigenome. So you can think of the DNA as hardware and the epigenome as software that tells the cell which genes to turn on or off to activate the protein responsible for its function and identity. And now, the most fascinating aspect of epigenetics is that unlike our genes, epigenetic marks are flexible, are dynamically remodeled by lifestyle factors, things like diet, environmental toxicant, and even emotions, stress. And so these factors collectively are described as the exposome. So all the things we are exposed to every day. And epigenetics explains these interactions between exposome and genetics, and why we know that our genes in general contribute only to 25% of our response to our complex traits, even chronic diseases in general. And I'm talking, of course, there are exceptions. There are monogenic diseases where you inherit a mutated gene and you have the disease. But for most traits and genetic variants, the environment has a bigger role. And the reason is that the environment talks to our genes through epigenetics. The link to longevity is that because epigenetics measures response to environmental factors, also changes with time. And in a way, it measures the passing of time. So that recently, in the past 10 years, so-called epigenetic clocks have been developed. These are tests that we can do with blood samples or saliva samples, and we can measure thousands of epigenetic changes and measure their association with so-called biological age. We used to think to our age in chronological terms. We celebrate our birthdays, and that is our chronological age. The biological age is an idea, a construct that expresses the very intuitive idea that we have that we may look younger or older than our age. And this looking younger or older is our biological age. So this is an intuitive concept. There is really not one single measure. We will never be able to measure biological age, but there are different ways scientists read this biological age. And one of them is by using these epigenetic clocks that measure the passing of time from the perspective of the epigenome.
So going a little bit deeper into that, I've taken a number of these tests where supposedly they're able to measure what your typical biological age would be and what mine is. And they did it through looking at something called biomarkers. And when I was interviewing Mark Hyman, he was telling me that not all biomarkers are the same. Can you explain what biomarkers are and how they can help give you some more insight into understanding what's working well for you and where you could put some more emphasis in these choices that you're talking about?
Yes, actually, I would love to also learn more about your experience and which kind of tests did you take. Because in order to reply to your question, I need to understand which kind of epigenetic clock you took. Because there are epigenetic clocks that are really just based on DNA methylation, which is one type of epigenetic modification that is used to build epigenetic clocks because it's the most easy, reliable, and accurate to measure, epigenetic modification to measure. So then there are other clocks, probably the one you took, where they just measure some biomarkers that can be blood biomarkers, your even blood lipids, C-reactive protein, and then they, based on the collection of this biomarker, they output a biological age. So these are not really epigenetic clocks, are functional clocks, are another way of measuring biological age. Remember, I mentioned biological age can be measured in different ways. There are three categories. So molecular clocks, they include epigenetic clocks, telomere clocks, And now we are looking at proteomic clocks, so molecules. Then you have functional clocks, where you measure biomarkers, for example, blood, lipid panels, but can also be digital clocks that measure your facial aging. And then there are also other tests like VO2 max, like other functional measurements. That's why I need to understand which kind of clock you took. And then there are, in the category of epigenetic clocks, there are different generations of clocks. The first generation is just a pure epigenetic measurement of different sites in your genome. The second generation estimates the values, the epigenetic values of functional biomarkers, like, for example, blood, lipids, or other protein, and then does an epigenetic score of those changes. So it's an epigenetic measurement, but it's an estimate of those markers. And then there are third generation clocks, where they look at the estimates of the changes in those biomarkers over time, so that the clock can measure the pace of aging, not only the age, but the pace of aging, because those estimate the changes in time. I know it's complicated. I can go over again, but it's just to say that your question probably needs to be decomposed, and in order for me to really answer the question, because things are more complicated than that. Yeah.
Well, I have used two primarily. One is Inside Tracker, and the way they did their testing is they did a blood sample, a saliva sample, and then had a whole listing of questions which were all about your lifestyle. And then I've also done Lifespan more recently, which is the company that was started by Dr. Peter Diamandis and Tony Robbins. And they did not do the saliva test, but they do a comprehensive blood test. And then they have lifestyle questions that they ask you as well. But Inside Tracker actually gives you a biological age report. Lifespan does not. They just tell you that across all your biomarkers, and they have some metabolic biomarkers, they have others that are mineral biomarkers, et cetera. They just tell you how much you've optimized. Are you 93 optimized, or are you 56% optimized? And then they try to tell you what you could do to bring those biomarkers up.
Yeah, I see. So I can't comment specifically on these two because I'm not familiar with them, but I can comment in general with the challenge of these clocks that, as you can see, there are so many of them. I think there are now over 20 on the market. And so as a practitioner, as a doctor, or a consumer, I would consider including them as a marker just without having the expectation that they will necessarily be precise or really able to guide any personalized protocol. I will take it more as because I like to advance the field and understand more and more data. So I think this is ideal for people who are excited by the science and want to be part of its advancement. But there are several problems. So as you can see, there are different companies measure the clocks in different ways first. Then I think that the major problem is that, as I told you, biological age is really a construct. So what this means is that I may be, if I take a digital clock, I may be older than my age because I love the sun. But because I'm exercising and I'm eating well, my muscle cells and my liver may be younger than my age. We don't have one biological age. We have a biological age for every single cell of our body. And even these epigenetic clocks most of them are based on your blood or saliva. And we know that, for example, These changes in blood epigenetics don't track the changes that are seen in brains of Alzheimer's disease patients. So the neurons are epigenetically older, but not the blood. What I mean is that, first of all, do not expect that for your condition, a biological test with epigenetics will be precise or will address your problem. And I think this is an important aspect. I didn't take an epigenetic test. It would be nice to, I'm more interested in functional biomarker tests, but I observe the field. It's very interesting. And so this is the first expectation. The second, very important, is the clinical. So if you take these tests, are you then able to give a personalized recommendation or to track the progress of an intervention? I'm not sure. Again, it depends. The challenge is that these clocks have been developed using databases of people that lived in the 50 years ago. So where also the environment was different, and they are true at the population level. What I mean by that is that if you take 10,000 people and measure their biological age with these clocks will be very precise. But if you take one person, could be off very much. It's not so, they work at the population level, not so much at the individual level. And one other thing is that not necessarily being younger epigenetically is we don't know whether this is necessarily a functional improvement. For example, we know that calorie restriction makes you younger epigenetically. But let's say I measure the epigenetic age of my elderly 82-year-old mom. She's a champion of longevity, but she's frail. She's frail. So if I measure her epigenetic age today, she goes on a calorie restrictive diets, and then after 1 month, her epigenetic age is reversed of 2 years. Hooray! But then she's more frail, and then she falls the day after and she dies. The point is, being epigenetically younger doesn't necessarily mean, or that is not an indication for a therapy. And then one last comment of caution. I know that I'm saying a lot of bad things. At the end, I will say good things about epigenetic clocks because I think it's an exciting field. But I need to say that because I'm very concerned. There are lots of studies now testing epigenetic clocks in the context of dietary interventions. You mentioned the study by Cara Fitzgerald. Cara is a colleague, and I also read her book and wrote a preface, and I had the opportunity to read the study, and she used a diet that was epigenetically very well designed. What I mean by that? The diet that Cara Fitzgerald used was a diet based on animal protein as a core and very rich in vegetables and plants. And why this is a very good epigenetic design? Because we know that the food we eat contains epinutrients. This is a word that I introduced and Cara is using it, and I'm very happy, which means there are nutrients that really nourish our epigenome. And animal protein is the richest source of one category of epinutrients called methyl donor. These methyl donors include things like vitamin B of the B group, so folate, 12, 6, and especially we can have only animal protein contains B12. And we know this, and we know, for example, a vegan need to supplement, but also choline. Choline is a semi-essential nutrient. We only produce 30% of the choline we need. And we need to get the rest from diet. The richest sources are eggs, shellfish, liver. And 99% of vegans are deficient. And choline is essential for production of acetylcholine in the brain for brain function and is an epinutrient. So animal protein is the foundation of an epigenetically complete diet. And then the plants, the veggies, provide other epigenetic epinutrient called epibioactives. Think of polyphenols in olive oil, in tea, coffee, but also things like curcumin, so spices that modulate the activity of enzymes that write and erase epigenetic modifications of our genes, regulating their function. And these epinutrients are very often present in things like in medicinal plants. And probably they explain why many plants have medicinal properties. Now, going back to the diet and the clocks. So Kara's diet was an epigenetically very well designed diet. And she showed that after 8 weeks of this intervention, the participants were rejuvenated by 2 years. Then there was a criticism to her study saying that after statistically correcting for the test and retest error of the epigenetic clocks, that difference was not significant. But then actually that statistical correction itself really fundamentally changes the clock. So just to say, it's complicated and you will find lots of people saying everything and the contrary of everything about epigenetic clocks. I am now participating in another study at Stanford where we tested epigenetic clocks in the context of a vegan diet. We didn't publish the results yet, but I will emphasize this in the discussion that whatever the outcome will be, we know that a vegan diet is not an epigenetically complete diet. So I encourage people not focusing on the results of the latest epigenetic clock study, even if the headline says vegan diet rejuvenate the epigenome. Go read the methods, because the reason, first of all, it's a short-term intervention. And then we know that nutrition studies are complicated. If you read the fine print, you will understand more and the complexity of the studies. In this particular study, this is an intervention with people who are overweight, and they go on a healthy vegan diet and a healthy omnivore. But if you look at the diets, there is a difference. The healthier omnivore is eating a lot of grain and starchy vegetables, whereas the vegan diet is eating only the non-starchy vegetables, and they are losing weight. This is just to say, I don't want you to be confused by the new science of epigenetic clocks so that you forget about the fundamentals of epigenetics and epinutrient. I call this epinutrition. So focusing on the biology of epigenetics and the nutrients that build it, and don't be distracted by the latest study, because you will see in the next years, you will find epigenetic clock studies supporting all kinds of diet: carnivore, vegan, plant-based, omnivore, and you will be confused just because this is the nature of nutrition science. Whatever diet you support, go on PubMed, the library of published research on the internet. You will find an article that supports that diet. And at the end, you will be confused. And it will be the same with epigenetic clocks. So don't do that. Just focus on biology and biochemistry, and then personalize this to your... I feel sorry for people because I am a scientist and I find this confusing. And I can only imagine how it is for people. Of course, they don't know what to do because every day they hear that this is good for you and or this is bad. And so let's go back to basics and then personalize these protocols for people.
Well, I just got a constant glucose monitoring system from Levels that I can't wait to try when I get back from an upcoming trip and start going down that path of seeing what that insulin and glucose mix on a daily level for me. And
Oh, yeah,
yeah,
that would
I'm
be.
excited about that. I know you've done a lot of studying of, and I'll mention this because you are from a Mediterranean based country, keto diet versus the Mediterranean diet. Can you share any insights from that?
Oh, yes. Okay. I will start by saying that when you read the headlines, always ask yourself, what is the keto diet or the Mediterranean diet that was tested in the study? Because, no, it's interesting. I made this comment when we were designing the study. The Mediterranean diet that we tested was a healthy Mediterranean diet. You know what? No refined grains. So I was born in Naples. I was eating pasta bread every day. And then the study tells me that the Mediterranean diet is a diet with no refined grains. So the point is, if you read the headlines and they say, Mediterranean diet is good for you, and then you think of your Mediterranean diet, you may think, okay, then I go eat pasta and bread. But actually, they tested their own version of Mediterranean diet. Our study tested a diet that was no pasta, no refined grains, no bread, and was already a very low carbohydrate, very healthy version of a Mediterranean diet, and compare that to a ketogenic diet. Again, was a very well designed ketogenic diet with less than 30 grams of carbohydrate per day. But it was a diet where, for example, was more heavy on meat and dairy than fish and olive oil. This is just to say that there are very different ways of doing a ketogenic diet. There is not, many people think that is always meat and butter, but it's not necessarily like that. I am on a low carb and maybe even keto. I don't know because I don't count but I am more fish and olive oil, and that's, you can do a keto Mediterranean. But anyway, the study was designed to compare these diets for pre-diabetic and type 2 diabetic patients and compare their outcomes on glycated hemoglobin, which is a standard marker for diagnosis diabetes, but also CGM, so continuous glucose monitoring, what you are now trying. And so you will be excited to know that both diets were really improved glycated hemoglobin. There was not a significant difference between the diet. But take into account that also the Mediterranean diet was very low in carbohydrate. Again, it was not a Mediterranean diet you think. But we also found that the CGM data were significantly better for the keto group. So again, and this is confirming what we have seen also in other trial from, you may be familiar with the Verta Health study, and it's now a multi-year study. I think now they are now in the 5th year, but I'm familiar with the results published after 2 years. This was an intervention using a ketogenic diet for diabetic patients, showing that all the markers were improved, and most importantly, most people were taken off medications. So basically, it's the diet, going on a keto diet can allow diabetic patient to go off medication. And this is really a food as medicine approach. It's about giving people options. I don't want to say everybody should be on a ketogenic diet, but I think the ketogenic diet is probably the prescription strength approach for diabetic patients. And I think that some people may not need or want to go to a prescription strength, but if the alternative is taking a medication, then at least they have the options. And I think that's where education is important. Say, look, going on a keto diet as a diabetic could allow you to get rid of medications. We have not seen this with studies with Mediterranean diet or more milder, with higher carbohydrate intake. And I think it makes sense from, a biochemistry perspective, because a ketogenic diet really allows our body to switch from using glucose to fat as a fuel and stabilizes glucose.
Well, I did want to ask you about one other thing. I had the opportunity to interview Dr. Gabrielle Lyon, who has a new book coming out, and she's known for being an expert on proteins. And you've talked a lot about the needs for good intake of proteins to support epigenetics. But another area she is also passionate about is muscle mass and the importance of doing strength training for longevity. Have you done any research in that area?
I didn't do any research, but as you can see, I am strong and I practice strength training regularly, and I've learned a lot by engaging in strength training. Actually, I started my strength training journey together with my doctorate. And that's where I experimented with the ketogenic diet at the beginning, just as a part of a performance program, because the ketogenic diet actually was popular in the 80s among bodybuilders who wanted to define muscles after a bulking phase. So I tried that. There were times in my life where I reached 10% fat and I was strong, and I still train, but now I don't cycle. I'm just on the same diet because I think I prefer to focus on health rather than performance. But just to go back to your question. So I don't have any research, but I have personal experience and I read a lot about the topic. Our muscles are really an organ that produces cytokines, hormones, molecules, and it's not only a structural component, it really transforms the way we metabolize and use food. And that's why personalization is so important. Even carbohydrates have a completely different effect on somebody who has a lot of muscles, because the muscle is really a sugar sink. It really claims sugar from your blood, especially after training. So I usually have my higher carbohydrate vegetables like pumpkin, and sometimes if I want to bulk up, even sweet potatoes after training, whereas I go with lower choices before training, because after training we have this phenomenon of non-insulin mediated glucose uptake, which is a kind of magic. Why? Because usually we need insulin to claim glucose in our cells. So we eat, insulin goes up and tells your cells, take glucose from the blood. But after training, we don't need it. It's our muscles that said, give it to me, give it to me. I need it because I burn my glycogen stores. This is just magic if you think about it. So if you eat that glucose after training, it goes to your muscles. Now, many bodybuilders, for these reasons, used to eat dextrose tablets just after training. I wouldn't do that, just because for me, I think it's so much better than to focus on nutrition. I have some healthy carbohydrates. But the point is, yes, muscle is important. Also, for longevity reasons, I hired a trainer for my mom, 82 years old. And I tell you this, not only she is stronger, she can walk alone, but she has so much more energy. And I notice that she just thinks more clearly and better. And she has had a transformation after starting weight training. And we know that there is even this muscle brain connection, and it's not only about movement, it's also about thinking and feeling. And so I believe that exercise, although I'm an advocate of diet, exercise is very important, and protein is very important, not only from an epigenetic standpoint, but also for muscle growth. I think is a very much neglected nutrient. There is a lot of controversy on this topic, but I think a high protein diet, especially a diet that contains some protein at each meal, is very important. It's not only about how much protein per day, per kilo. Yes, these are quantities just to give us a guideline, but how much protein per meal? Because we don't have a place to store protein. We have a place to store fat, adipose tissue, and glucose, glycogen, not protein. So that's why we should also always be eating protein, some protein, probably 30 to 40 grams every meal.
Well, thank you for that, Lucia. And I understand you've got an upcoming course that some of the listeners may be interested in hearing about.
Yes. So thank you for mentioning that. Yes, I've been teaching epigenetics for many years at Stanford. I'm proud to have introduced for the first time epigenetics in the Stanford Genomic Certificate. This is a professional program. So I decided to update those teachings and distill them in 5 modules for all the nutrition science enthusiasts out there and make it accessible. So you can find the course at draronica.com and use a coupon code PASSION50 for a 50% discount.
Well, thank you, Lucia, for sharing that. And thank you so much for joining us today. It was such an interesting deep dive, especially into the nutrition behind epigenetics.
Thank you very much, John, for having me. I hope your audience enjoyed our conversation and be inspired and use food and exercise as medicine.
I thoroughly enjoyed that interview with Dr. Lucia Aronica, and I wanted to thank Lucia and Dr. Kara Fitzgerald for having her appear on today's show. You're about to hear a preview of the Passion Struck podcast interview that I did with Dr. Richard Ryan, a clinical psychologist and co-founder and co-developer of the self-determination theory, an internationally recognized leading theory on motivation. Dr. Ryan lectures frequently in the U.S. and abroad and was one of the most cited researchers in the world on both psychological and behavioral functioning. We are going to explore in depth the topic of self-determination theory. In that episode, we do a deep dive on the connections of many things that I talk about on Passion Struck and self-determination theory. One of my best interviews yet.
Recent research in neuropsychology has really been showing that we do have a set of internally rewarding experiences that are behind a lot of our activities. When people are doing something benevolent, when they're doing kind things for other people, they're also feeling striatal activation. They're showing activation in the reward centers of their brain because something satisfying has occurred for them. I think in this way, it's very much supported the general tenets of self-determination theory.
Remember that we rise by lifting others, so share this show with those that you love and care about. If you find someone who's interested in understanding epigenetics, then definitely share this show with those that you love and care about. In the meantime, do your best to apply what you hear on the show so that you can live what you listen. Now, go out there and become passion struck.